Okay, we're starting something new here. Moving into the cytoskeleton. It's kind of a little intermediate unit. It's always been one of those units that's hard to figure out where exactly you could put it in the course, because you could pretty much stick it anywhere because it's self-contained, but it ties in with a lot of other cellular processes. So as you guys know, the cytoskeleton basically is comprised of three separate component systems that are most easily separated just based on the sizes of the skeletal components. So the smallest being the microfilaments that are comprised of the protein actin. There's these guys in between that, due to a lack of a better name, they call them intermediate filaments. And then we've got the biggest guys, and those are the microtubules. And they all work together. There's a division of labor among the three cytoskeletal components. They each have their own responsibilities. But in many cellular processes, they actually have to function together. And where you're really going to see that come together is in a few weeks when we talk about mitosis. Okay? So one of the things to understand is the cytoskeleton has the ability to be static in many, many cells. So for example, erythrocytes, they're all just kind of typically hanging around. An erythrocyte pretty much always looks like an erythrocyte all the time. So the cytoskeleton there is not particularly dynamic. There isn't much going on. But in many, many other cells, even when the cell itself looks like it's not doing anything, the cytoskeleton within it is actually extremely dynamic. And so what we're going to be looking at in this unit is we're going to focus on features of microfilaments first and then microtubules. And more or less just focus on, initially, what makes their structures dynamic. How do we change their stability? And in the case of microfilaments, we're going to be doing that to look at how cells themselves move, cellular motility. So how do we change microfilament cytoskeleton to allow a cell to move in a directed manner? Microtubules, we're not going to focus so much on the dynamic part of it as we are how microtubules are used as an infrastructure to allow trafficking to take place within the cell. It kind of just extends what we just finished with vesicle trafficking. Because all of that movement that you see of vesicles moving throughout the cell occurs on the microtubule cytoskeleton. Okay? So, and we're going to come back to all of them again, like I said, when we get to mitosis in a few weeks. So we're going to start off with the simplest system, and those are microfilaments. Okay? The underlying protein that we'll talk about is the protein actin, globular protein. As you're going to see, it's one of the most abundant proteins in our cells. It's a nucleotide binding protein. And the nucleotide that's involved here is ATP. And in fact, actin is an ATPase. And what you're going to see is that there's an exchange factor that's going to be involved that's going to alter microfilament dynamics by promoting the exchange of ADP for ATP. Okay? Microfilaments, as you're going to see, have polarity. The two ends differ from each other. Okay? And that is going to influence growth and shrinkage. That is, the two ends are not equal. All right? Now, microfilaments, their main role, if you take a look up here, is it really deals with cellular architecture, three-dimensional structure. So, for example, in the intestinal epithelial cell that we talked about earlier in the transport section, microfilaments are responsible for forming the microvilli. We bundle a bunch of them together. And they extrude the plasma membrane up here at the apical membrane so that we can increase the surface area. They're also involved in elaborating a structure called the cortex, which goes around the basolateral membranes and gives it rigidity, gives it strength. And then we have the adherence belt that basically seals off the apical membrane from the basolateral membrane. So they have a real function here in cellular integrity. Where we're going to be looking at them as well, though, is down here when we're going to be dealing with cellular migration. And that is the process by which a cell is going to respond to an extracellular signal. And the cell is going to want to migrate towards it. So the issue is, how does the cell perceive the signals out there? And then secondly, how does it redirect microfilament polymerization so that they literally push the plasma membrane towards the source of the signal? So that's where cell motility is going to come in and have a major function. Okay. So, motility is going to be the important part here. And what you're going to see is it links signal transduction with the cytoskeleton. So this is one of these reasons why where you put the cytoskeleton is kind of awkward in the course. Do you talk about signaling first and then talk about the cytoskeleton, or do the cytoskeleton first? We're doing it that way. So we're not going to focus so much right now. We'll move into it after this unit on the mechanism by which extracellular signals bind to integral membrane protein receptors and initiate a signal transduction process. Okay? We'll get into that, and we'll look at it in terms of changing cellular physiology. So for right now, ignore the mechanism of the signal binding to the receptor. We're going to do an end run on that right now. And instead, what we're going to focus on is the fact that there are lipid-anchored proteins, small G proteins. It's just an extension of the ones we already talked about, like RABs. And these are called the Rho class G proteins. Rho, because Rho was the prototype, it was the first one to discover. These small G proteins are lipid-anchored in the plasma membrane. Like every self-respecting G protein, their inactive form is going to require that they have GDP bound. Okay? So that means that in response to the binding of an extracellular signal to its receptor, we have to recruit a GEF. The GEF comes in, swaps out GDP for GTP, and it's going to be the activated form of one of three small G proteins. They're just showing Rho right now. The GTP-bound form becomes a binding site to nucleate the formation of microfilaments. And what happens is that it's very simple. Since the G protein is lipid anchored to the membrane, and it's only going to be activated in close proximity to where a receptor binds to an extracellular signal, microfilaments are literally going to polymerize against the G protein and push it. They're going to physically push, then, this whole region of the plasma membrane by nucleating their formation here where this particular G protein is activated. Okay? Now, let me give you an idea of how quickly this takes place and how reversible it is. Here's an example of chemotaxis. Right? This is a cell, it's an amoeba, that's being perversely stimulated by an investigator who's basically got a pipette here that's dripping out a concentration of an extracellular signal. You move it around, comes over here, comes down, move it over here, and you can keep this up as long as you want to keep torturing this poor cell. But here's the point. If you take a look at this, notice that the cell is moving in a directed manner towards the signal. This is what's driving that. So in response to the activation of receptors by a signal that are causing localized activation of a G protein, what's causing that rapid movement is the active microfilaments are literally polymerizing and pushing only the plasma membrane where the signal is coming in. All right? So any sort of chemotactic response, macrophages moving towards the site of a pathogen, involves this particular process. It's rapid and it's reversible. Here's another example that we're going to be talking about. And this has to do with the response of a cell to a gap. I mean, a gap meaning the absence of other cells immediately touching it. It happens when you have a wound. If you cut yourself, all right? Extracellular growth factors, for example, platelets are going to release clotting factors when you cut yourself. Platelets are also releasing growth factors. And the growth factors are necessary for two responses. The short-term response is to trigger the cells adjacent to the wound to migrate into it, to literally close it up and fill it in. The longer-term effect of these growth factors is to cause those cells to re-enter the cell cycle and to proliferate, to undergo cell division. But that takes several hours, many hours to take place. The short run is to basically start filling in the wound by promoting cell migration. So the way that you can see that here is that here's a fibroblast, a migrating cell. And what you're looking at here is that these are cells that are expressing GFP fused to actin. So you're looking at actin now hooked up to be fluorescent. And now what you can see is that there's a growth factor being produced over here, and the cell is migrating towards it. Now what you notice here is that the migration is occurring on a very, very wide front. The cell is not just advancing in a little narrow band. So one of the things that you have to do when we're reconfiguring the cytoskeleton for cell migration here is that we can't just make long linear microfilaments that are projecting individually against the plasma membrane like spokes on a wheel. Instead, what we have to do is have a mechanism where the microfilaments can branch out. And by doing that, they can then project a larger surface area of the plasma membrane and get the cell to advance over a much, much broader front. And so you're going to see that depending on the nature of the G protein that's activated up here, we can cause linear microfilaments to grow, or we can cause branched ones to grow. The other thing that's equally important is that the cell can't get too far ahead of itself. This guy over here, and this one over here. And that is, well, what is going to be referred to as the leading edge of the cell moves forward. Well, what about the back of the cell? The trailing edge. It can't just leave it there, because as you guys can imagine what's going to happen, the cell's going to stretch itself out to such a point it would snap. So while we have these events that are taking place up at the leading edge of the cell, that's moving in the direction of the signal, the trailing edge of the cell is going to require that the microfilaments depolymerize and fragment so that the back half of the cell can literally pick itself up and contract and move forward and keep up with the front edge of the cell. So those two processes are going to be happening simultaneously. Okay? All right. So let's take a look at the main player. The protein that we're going to be dealing with is actin. Okay? It's one of the most ancient proteins out there. It was one of the very first proteins to evolve because as soon as you started to put together a cell with a contiguous plasma membrane, you needed something to give it some integrity, which is more than we can say for one of the figures up here. It's the most abundant protein in our cells. It makes up 10% of the weight of the total protein in your muscle cells, so that's how abundant it is. It arose from a bacterial ancestral protein, so bacteria do have a cytoskeleton, and it's comprised of a protein called m4a. Okay? So it's an ancient protein. It's changed very little over evolutionary time. And I know this is an insult to the amoeba, but if you compare the amino acid sequence of a human to an amoeba, the amino acid sequences themselves are 80% identical. I'm not talking about conservative substitutions. I'm talking about identity. So this is a protein that evolved very rapidly, very, very quickly, adapted the ability to function very, very, very efficiently in a very, very wide number of physiological contexts, and it has stayed that way. It's one of those proteins that cells got right from the very beginning And there's been very, very little need to adapt it or modify it over subsequent evolutionary time. Okay? It's a good protein. It's a relatively simple protein. It's the prototypical globular protein. It's very compact. You can divide it into 4 domains. It's kind of got this little leafy structure here, a little bit like a 4-leaf clover. And inside is the ATP binding site. Now you notice on the figure it also shows a magnesium ion. And just something to point out is that any time you've got a nucleotide binding protein, like actin, or even any of the G proteins, the nucleotides are never free. The phosphate is always coordinated with a metal ion, and generally that metal ion is always magnesium. We usually just ignore it. We don't even show it. We don't diagram it. We don't list it when we say, for example, RAB GTP. It's just always there. Okay, so the phosphate's always coordinated to a magnesium ion. They're just happening to show that here for the structure of actin. All right? One thing you notice is that the molecule itself is not symmetrical. And what it has up here is a little cleft. It's an opening. And it has that little cleft so that ATP can bind to it, that it can be hydrolyzed from ATP to ADP, and that the phosphate or the ADP can diffuse away and be replaced with ATP. So the important point here is that the ATP is accessible through this little cleft. But equally importantly is that it means that the starting point to make a microfilament has polarity. Actin itself is a polar molecule. All right? The way that we're going to refer to this, the monomeric actin, is we call it G-actin for globular actin. That's just free actin hanging out in the cell. When we polymerize it into a microfilament that we'll abbreviate as MF, we also refer to this as F-actin, filamentous actin. The polymerization of actin into microfilaments does not require ATP hydrolysis, but it occurs. Okay? So ATP hydrolysis is not required for polymerization, but as you're going to see, it's going to influence the kinetics of polymerization. And that is what you're going to see is ATP hydrolysis occurs after G-actin polymerizes into microfilaments. And what this means is you're going to be able to separate the 2 ends of a microfilament from each other because one is going to have what's referred to as an ATP cap. The other is going to have an ADP cap. And that's how we're going to be able to tell them apart. Okay? All right. So if you take a look at a microfilament, it polymerizes. It actually makes a little double helix. It's a right-handed double helix where the microfilaments wrap around each other. Here are the 2 ends. I don't want to hear the word positive or negative in here, even though some of you guys did very, very positively well on the exam. Some of you a little negative, but most of you are very positive. But we're not going to use that term. This is the plus end. That's the minus end. It's not a battery. It's not a charge. So don't use positive, negative. It's plus end, minus end. Notice if you look at the plus end down here, the ATP cleft is embedded. Okay? So let me just flick back to this previous slide, okay? This would be the plus end down here. This is the minus end up there, where the cleft is going to be exposed. So the plus end, there's the cleft. So that means that this actin adds on right here, or this one added on here, the minus end or the cleft end adds on to the plus end. So they're all lined up head to tail. You go down to the end of the minus end, and it's the minus end or the ATP cleft that is exposed. Okay? So you can see that the structure's got polarity. the actin is not organized in there at random. They all have this particular alignment. Now, there's a cute trick that we can use experimentally to distinguish the plus end from the minus end. And that is, we can take the motor protein myosin. Myosin is the motor that normally causes actin microfilaments to slide past one another. We use it in muscle contraction. If you cut the legs off myosin so that it can't move, but only the heads can bind to the microfilaments, you get this unusual what's called decoration. And that is, you can decorate microfilaments with myosin heads. And the myosin's heads form this really interesting arrangement where they line up like arrowheads. So these are like the heads, this is the arrowheads. These are all myosin heads. No one really knows why it binds to actin this way. It really doesn't matter. But the neat thing is, they're all pointing towards the minus end. So if you're ever looking at a myosin-decorated microfilament, all the arrows point to the minus end. The barbed end are always corresponding to the plus end. Okay? So it's just a way that you could visually take a look at a microfilament and go, which end am I looking at? Okay? All right. How do we make them? Okay. One of the things to understand when we talk about microfilaments and microtubule dynamics here is we need to make a distinction between what we're going to do in a cell-free system that has nothing more than purified actin or purified tubulin when we get to it. The dynamics are going to be different in a purified system than it will be inside of a cell. Okay? So these initial experiments that we're looking at here are looking at what governs the polymerization of actin in a purified cell-free system. Okay? So here's what you notice. You've got a tube full of actin ATP. And what you look at is that you put it into solution, and we're starting off with this. And you notice that if you look at what's the time course, what are the kinetics that it takes to convert G-actin to microfilaments? The first thing you notice is there's a lag. And that is, there's a rate-limiting step that has to take place to take free actin and convert it to microfilaments. And the rate-limiting step is the formation of a nucleus. It's exactly analogous to seeding a crystal. When you guys do crystallizations in Orgo Lab, you have to get enough of the compound to come together, the solute, to come out of solution, to basically grow a crystal. And the rate-limiting step is seeding that crystal. You need a nucleus. The same thing happens here. So the rate-limiting step, when you've got purified actin, to get it to turn into a microfilament, is to get about 3 G-actins to come together, and that makes a nucleus. And you notice that once that forms, now it grows very, very rapidly. Okay? You notice it plateaus at steady state. Once you hit steady state, there's no more net further conversion of actin to microfilaments. And the reason for it is you've run out of free actin. Okay? It's basically now you're in equilibrium between a microfilament dissociating and then forming a new one. So here's something you can do. If you take a look at the kinetics here, we can speed it up if instead of starting the reaction with just pure actin, if we start it with nuclei, and that is we seed the reaction from the very, very beginning. And you go, well, where can we come up with nuclei? Well, we can take an existing microfilament and we can cut it into little pieces. As you're going to see, there's an enzyme that will actually do that. So now what we're doing in this reaction is that we're going to spike it at the very, very beginning with little fragments of microfilaments. And that provides the nuclei. And you notice that now when we add these to our in vitro reaction, so now we have little pieces of microfilaments. And now we add purified G-actin. You notice we immediately get polymerization. Our cells don't ever have to worry about this problem. Okay, this is an in vitro artifact. When we're talking about what occurs in our cells always have a pool of microfilaments that can always be used as a source of nuclei. So inside our cells, we never go through this rate-limiting step here. Instead, we're always going to be starting by growing or shrinking existing microfilaments that are going to be used as a source of nuclei. So what it means is this. You're always recycling your microfilaments, breaking them down to make new ones. The other thing that you're going to see is that there's two ways we can make a microfilament shorter. One way is we can depolymerize a microfilament by having individual actin molecules dissociate from either end. They can fall off one at a time. That's slow. The quicker way to shorten a microfilament is to cut it into little pieces. So as you're going to see, if you really want to get rid of a microfilament, the fastest way you can, chop it into small fragments and it'll collapse. Okay? So you're going to see that both of those are going to be used inside of cells. Okay. How do we grow a microfilament? The two ends are not created equal. That's number one. Number two is there's a concept that we have to introduce that's going to describe the dynamics here that's called the critical concentration. Okay? And that is now, instead of looking at a time course of conversion of G-actin to F-actin, we're looking at the concentration dependence. So now on the x-axis, what we're looking at is we're increasing the concentration of G-actin in the reaction. And on the y-axis, we're looking at the mass of either monomers, which is G-actin, or microfilaments, which is F-actin. So what you're going to do is that in this experiment, you're starting with increasing concentrations of G-actin. So you notice that all the way down here, you're adding more and more G-actin to the reaction. It's not being converted to microfilaments. It's just you're accumulating, here's more G-actin, more G-actin, more G-actin, more G-actin, bang. All of a sudden now, you hit what's referred to as the critical concentration. The critical concentration is the concentration you need to now start polymerizing. So if the concentration of actin is below the critical concentration, you won't convert any of it into microfilaments. But once you attain the critical concentration, now the monomeric actin that you're putting in gets converted to microfilaments. And now you will start to grow. Okay? Here's where it gets complicated. The two ends of a microfilament don't have the same critical concentration. The plus end grows 12 times faster than the minus end. So there's a bias that's intrinsically built into microfilaments that says, grow at the plus end, don't grow at the minus end. They have different affinities to bind actin. So here's what's going on. If we take a look at a microfilament here, here's the plus end, here's the minus end. So you've got an existing microfilament here, here's free actin. Which end are you going to add on to? You're going to add on to the plus end. It has a much higher affinity to bind to the plus end than the minus end. So here's where you start getting a difference in the structure of a microfilament as you go down its length. Okay? Remember that the substrate that goes in for G-actin has ATP bound to it. So what it means is this. We take this actin, we add it, it's got ATP. We take another one and we add it, it's got ATP. So what you're seeing down here is at the plus end, all of the actins that you just added still have ATP bound to them. Over time, that ATP hydrolyzes to ADP. So as we work our way down the length of a microfilament, here's actin that added a while back. Okay? This used to be the plus end, but now we've added more onto it. What you see is we work our way down the length of a microfilament. We no longer have ATP, but instead we have ADP. But in fact, the phosphate hasn't diffused away yet. It's still there. It hasn't floated away from the microfilament. If we work our way even further down towards the minus end of this original microfilament, now the phosphate is diffused away, and now what we have left with down here at the minus end of the microfilament is ADP actin. It's a gradient. So now, here's what this means. If you're actin, and you've got a choice between do I want to add to the minus end or do I want to add to the plus end, you can now see that ATP actin has a much higher affinity, a lower critical concentration to bind to the plus end than it does the minus end. The critical concentration for ATP actin to bind to the plus end, the critical concentration is only 0.12 micromolar. It's really low. The critical concentration for it to add to the minus end down here, to take it and add it here where it's going to have ADP, is 0.6 micromolar. Okay? Not only that, but the rate of addition here, the rate with which this adds, is about 12 times faster than it is down at the minus end. So here's the point. Microfilaments are designed with an intrinsic bias to grow at the plus end. Okay? That's where they prefer to grow. The cell is going to take that bias and exaggerate it. They're going to set it up in such a way that within your cells, pretty much all of the action is going to take place at the plus ends. Okay? Now, so what does it take to make a microfilament grow at both ends? Well, it means the actin concentration is going to have to be above 0.6 micromolar. You're going to have to get actin above that concentration to get growth at both the plus and the minus end. But even if you're above 0.6 micromolar, it's still going to grow 12 times faster at the plus end than it will at the minus end. Okay? Now, what happens if we start dialing down the concentration of actin? What if we're in between? What if we're above the critical concentration? This is all in vitro. What if we're above the critical concentration for the plus end, but we're below the critical concentration of the minus end? What happens? Can we grow? And the answer is yeah. We grow at the plus end, but it shrinks at the minus end. Okay? So one of the interesting things about microfilaments is, if you're not growing, you're shrinking. And this is a process that our cells can use to just kind of keep a microfilament at the same length. So we're not making it longer, we're not making it shorter, but it's a very dynamic process. And that is something that's happening at each end. And it's a process called treadmilling. So when microfilaments are treadmilling, we're adding G-actin here at the plus end. But what's happening down here at the minus end is every time we add one here, we lose one down at that end. So if these blue actins here were at the original plus end, can you guys see that what happens is they actually work their way down to where eventually they're going to become the minus end? Because this guy started here, we add two more onto it, we add several more onto it, but every time we do that, somebody falls off down here. So eventually the actin that started off at the plus end winds up being down at the minus end and will ultimately fall off. So under conditions of treadmilling, it's a very dynamic process. Grow, shrink, grow, shrink, grow, shrink, but you have no change in net length. Okay? Now, this is pretty much everything that's happening in vitro. Now we've got to shift it and say, what goes on in our cells? To start with this little bias, and how can we exaggerate it? So what are cells going to do to alter this basic process? First thing that we can do is basically control polymerization under conditions where the concentration of actin is always going to be above the critical concentration. So here's the first dilemma your cells face. The concentration of actin in your cells is ginormous. And remember, it's the most abundant protein in your cell. The concentration is always going to be 50 to 200 micromolar. Remember, here's the concentration, the critical concentrations at each end. It's way above that. So theoretically, you should have no unpolymerized actin in your cells. All of the actin inside your cells should be completely polymerized. It should all exist as microfilaments. But it doesn't, okay? It turns out that just under half of it, 40% of the actin in your cells aren't polymerized. So how can cells control this when the concentration of actin is always going to be above the critical concentration? And there's two mechanisms that they have available. One is to make the ends of a microfilament disappear. They can make them invisible. The other is to make actin invisible. And it requires using other proteins. So the first set of proteins that we can bring in are going to be capping proteins. So let's say we don't want to grow the plus end, even though we're going to be above the critical concentration. We don't want to grow there. Our cells have a protein called CAPZ. And what CAPZ does is when you add it to the plus end of a microfilament, the plus end is now invisible. We can't add anything to it. It's just frozen there. Nothing can be added to it. So that's one way the cell can prevent polymerization at the plus end, even though the actin concentration is way, way, way above the critical concentration. So this is a plus end capping protein, makes the plus end go away. Well, what about the minus end? What if we don't want anything to happen down at the minus end? Either polymerization or depolymerization. Our cells have a minus end capping protein that they use. And that protein is called tropomodulin. So tropomodulin makes the minus end of a microfilament go away. It also keeps it stable. So now you can see what cells are doing is that they bring in these other proteins and they can basically modulate or control, do I grow or do I shrink at the plus end or the minus end, but now I don't have to worry about the total concentration. Okay? So it's a very simple mechanism to allow that to happen. Here's the other way that they do it, and that is they control the accessibility of free actin in the cells. So actin itself is constantly cycling between different pools. Okay? We'll start off up here. So basically, here's a microfilament in the cell. Now remember, we need nuclei in our cells to grow new microfilaments, okay? The way that we're going to use that is using this protein called cofilin. Cofilin binds to your microfilaments and chops them into little pieces. That does two things. It gets rid of that microfilament there very quickly, fragments it into little tiny bits. These little fragments are ADP actin. They are going to depolymerize into monomeric actin very quickly. So what we're doing here is we're recycling what was in a microfilament to generate more monomeric actin. But can you guys see that the monomeric actin that you're generating here has ADP bound? We need to exchange it back to ATP if we want to give it a high affinity to bind to and form a new microfilament. So here's the protein that's going to do that. The name of the protein tells you what it does. It's called profilin. It promotes filamentization. Profilin is an exchange factor. It does the same thing that a GEF does for a G protein. And what profilin does is it promotes the very, very rapid exchange of ADP for ATP. So the first thing that it does is it regenerates a pool of ATP actin. So now that's ready to polymerize. But profilin does something in addition to that. And that is profilin does not dissociate from the ATP actin. Profilin remains bound to it. And here's what profilin does. Profilin changes the specificity of actin to where profilin actin can only add to the plus end. So remember, actin already has a pretty high bias to polymerize the plus end. This exacerbates it. So profilin ATP actin can only add to the plus end. Now you can't grow the minus end at all as a consequence of having this protein. There's one more component in the cycle we need to address. And it's this protein here called thymosin beta 4. Thymosin beta 4 does the opposite. What thymosin beta 4 does is it binds ATP actin and makes it invisible. In essence, what it does, it takes ATP actin out of the available pool to grow into a microfilament. Okay? So the key thing is this, guys. These three proteins play critical roles in controlling the availability of ATP actin, and they control the ability of ATP actin to polymerize or not. Okay? So just to review them, cofilin gives you fragments, chops an existing microfilament down very quickly. That's going to play a major role, as you'll see on Monday, in how the trailing edge of a cell retracts and keeps up with the leading edge. That's the first point. Second point is, profilin recycles that ADP actin, turns it into ATP actin, but now enables it only to add to the plus end. And as you're going to see on Monday when we look at movement, all the action takes place at the plus end. That's what we're going to be doing, is recruiting profilin ATP actin just to the plus end to get a cell to move just on that side. And then thymosin beta 4 makes actin invisible. Okay? So the last point for this morning. Profilin has another function. And that is, there is a genetic form of ALS, for amyloidotrophic sclerosis. Many of you may have heard of this disease in the past. If you're not American, you probably never would have heard of it described this way anyway. And that is, ALS was more commonly known at one time, particularly in the United States, as Lou Gehrig's disease. Okay? Lou Gehrig was the first baseman for the New York Yankees back in the 1920s, the same team. They kept winning the World Series over and over and over. He was on the same team with Babe Ruth. But nowadays you say Lou Gehrig to most people and they go, huh? But Lou Gehrig was one of the first celebrities who came down with ALS. ALS is a neurodegenerative disease. It basically causes the complete loss of your motor neuron muscle function. Okay? You basically waste away. Eventually, it causes paralysis and death at a relatively young age. There's different forms of ALS. There's different causative agents of ALS. They're protein misaggregation diseases. And that is, different types of ALS can be precipitated, literally, by proteins having missense mutations that cause them to misfold and aggregate. There's one cohort of individuals who have ALS because of a missense mutation in the profilin gene. Okay? It turns out it's a significant number of them. It's about 10% of familial ALS. It turns out the missense mutation affects profilin. And what it does is that the missense mutation in profilin causes it to aggregate inside of neurons, and it basically starts to cause the protein to aggregate and accumulate. When that happens, profilin can't do its recycling function. So the problem is that in ALS cells, the missense, misfolded form of profilin blocks this cycle here. And what it does within neurons, it eliminates their ability to basically cause the cells to participate in axon guidance. So what it basically does is it causes neurons to no longer be able to form appropriate synapses, and they actually start to degenerate at the axon terminus. And that ultimately is going to result in de-innervation of muscle cells, and that's what causes the paralysis and death. So the point is that profilin is an important protein, not only because of its cycling function, but also because it's prone to these misaggregation folding disorders. Okay? All right. So that's a wrap for today. We'll have a homework set out on this afternoon. I'll get the exam scores posted this afternoon with the answers key so you can see how that went. Okay?